Hello everyone, I'm Natalia Falagán. I'm a senior lecturer in food science and technology at Cranfield University. So I'm going to try to explain a little bit what we do. It's probably a little bit different to the fundamental pure molecular science that you might have in your mind when you think about biology or a career in plant sciences. What we do in my lab is looking at post-harvest of fruit and vegetables mostly. Everything that happens from the point of harvest. Sometimes, traditionally, there's been lots of interest around how fruit and vegetables grow, how they interact with soil, the environment, but kind of it stops. That research kind of sometimes stops once the food is harvested. However, there's lots to do afterwards, and I'm going to try to bring you a case study here, a little bit of what we do in my lab, and yeah, I'm happy to answer any questions later on or tomorrow that I will be with you too. I work in the plant science laboratory at Cranfield University, which is a postgraduate-only university. We have only master's degrees and PhDs, so that's something we are specialized in that. Which means that our links with industry and our links with research councils is really, really strong. And we have a really good balance between that teaching and our science. As I said, we look in our lab from harvest, so we have facilities like these rooms that are cold rooms. We can change temperature, we can change gases, we can change humidity, and try to understand that post-harvest response to those changes that happen in the supply chain. Once that's happened, we take it to our labs. We analyze the quality and safety of fruit and vegetables at three main levels. Physiological, that is what you would see as a consumer. If it looks, the appearance is good, the color is good, the taste is good or bad. And biochemical, so all the nutritional traits that are behind that food that you're eating. And then to really understand who's regulating all that and who is responsible for those changes, then we go deeper into the molecular level. In this context, my interest in, or our interest in post-harvest is crucial because at the moment we have global food systems that are really, really complex. Our fruit and vegetables are the food category that are the most perishable, that are presenting the most losses, and also they are very nutritious, so it's important that we keep them as healthy as possible until it reaches the consumer. We have an increasing food demand. I know one of the speakers have been talking about this morning, so it kind of links to that talk. We have high volumes of food loss and waste. And we have issues around energy supply. So our global temperature is increasing. We have to keep fruit and vegetables at low temperatures. That's not really possible because of prices, because of affordability, accessibility to energy systems that are sustainable and resilient, and that increases our food loss and waste. And on top of that, we have competing resources. We have soils that are being degraded, that affects the post-harvest quality of fruit and vegetables. We have water resources that are limited and also lower in quality than before. Again, it affects the quality, nutritional and physiological organoleptic quality of our fruit and vegetables. And then we don't have enough people that are specialists in post-harvest biology and technology to be able to help us tackle these global challenges. At the end of the day, what we are trying to do in our lab, with that base in biology, is to create or help create resilient and sustainable food supply chain at a global level. As I was saying, you will see these. The fruit and vegetables are very, very sensitive to any changes. and it's important that we understand the physiological mechanisms behind their post-harvest behavior, so we can develop technology and strategies that are able to maintain that quality from harvest until it reaches the consumer. And that can look like in very different shapes and forms. It can relate to temperature only. It can look at packaging, edible coatings, gaseous environment, that is what I'm going to talk about today, and any other form that you can think of as the future leaders of plant sciences. At the end of the day, what we are trying to do is reduce that 40%, 1/3, it depends on the country, it depends on the supply chain, that is lost across the supply chain. If we look at countries like the UK, we don't have that many losses pre-harvest. We have really good infrastructure that is able to avoid those losses before harvest, but then we have the issues post-harvest, where when the quality is not achieved based on the consumer demand or retail demands, there are losses there. So we have to think about that. If we go to low-income countries, it's more or less the same proportion, but it's pre-harvest or pre-consumer, where we don't have enough infrastructure for cold chain, when we don't have enough knowledge on when to harvest or how to handle the fruit and vegetables. So the proportions are there, and we try to touch on all this. One of the strategies for storage that we have in post-harvest is the use of controlled atmospheres. I'm not sure if any of you have heard of this before. I hope so. The idea behind this strategy that started in the 50s, 60s, is to reduce the level of oxygen in the surrounding atmosphere of the fresh produce and increase the levels of CO2. We have 0.03% of CO2 in our atmosphere and 21% of oxygen in our atmosphere. So I'm talking about reducing oxygen down to 5-6% and increase CO2, carbon dioxide, from 0.03 to 10, 15, depends on the product. Each product and each variety, each cultivar, has a different requirement. So you can, this is not one fits all, okay? So this is important to know. What we do is changing that environment, and what happens is that the respiration rate of our fruit and vegetables slow down. Yes, fruit and vegetables respire. So what they do is basically consuming their own resources, because when they are in the tree, they have all that resources coming from the tree or the bush or the plant. But when they are on their own, they are consuming their own sugars, their own organic acids. If we manage to slow down those degrading processes that are mostly regulated by respiration, we are going to avoid that loss in nutrients or delay. We are going to maintain a better structure of our tissues, maintaining a better firmness, and then have a better appearance and less impact around losing that, compromising the integrity of that tissue that can lead then to decay and spoilage. Okay? The issue, this has been applied for years and years, and it works really well. The issue is that if suddenly I change the oxygen concentration, for example, from 21 to 5%, it creates a metabolic shock that affects the quality of the fruit or vegetable, the fresh produce. It's like if I took you and I put you in a room at 5% of oxygen, you'll probably get very stressed out because you don't have enough oxygen to keep on breathing. And you will probably start breathing really slowly, like something is going on here. So, and you will probably slow down also your metabolism. You might die, but that's not the case of fruit and vegetables, okay? So, but it still affects their quality. So what we thought is to apply a new approach to control atmosphere called graduated control atmosphere. And what we do is decrease the concentration of oxygen and increase the concentration of CO2 gradually, okay? Instead of immediately we take 2, 3, 4, 5 days, depending on the fresh produce, to do that. What we try to achieve with this is help the plant, or in this case the fresh produce, to adapt to that changing environment and not have that metabolic shock that happens if it changes suddenly. So we did this experiment that is published now in 2020, I think, and we had different treatments: control, that is normal air; control atmosphere, that is that sudden change in the environment that went down to 5% of oxygen, 10% of CO2, and then the 2 gradual control atmospheres, one that went down to those final concentrations in 7 days, and another one in 3 days. Okay? We maintained the same temperature, 0 degrees, that is the storage temperature for blueberries. All this was done in blueberries, sorry. And 90-95% of relative humidity during 30 days of storage. And what we have to do that is a gas mixer. So we replace oxygen with nitrogen, and CO2, we just increase the CO2. And we had like different reps, and we measure, for example, respiration rate with a respirometry system that basically measures respiration real time. What we saw was quite interesting, at least for me. So we did it during 2 seasons. We normally try to work with different seasons, not just one harvest, because there are other pre-harvest factors that can affect the results. So we try to see it in different harvests normally in this field. And we saw this is the concentration of CO2 that the fresh produce is respiring. You normally produce CO2 and absorb oxygen. The peak here, the highest metabolic stress is showing, okay? So if you see the red line in both years, that's the traditional control atmosphere. It peaks really high because they are perceiving that there is very low oxygen. It's followed by the blue line. The blue line is the one that achieves that change in 3 days. So it was also very stressed out. Then we have air that has this peak because of the change in temperature exclusively. And then lower down, we have the one that changes the environment in 7 days. So that's an interesting point here. And the second point is that both graduated control atmosphere, the red and the blue, the green and the blue, mostly the green, achieve the lowest respiration rate during storage. This means that was respiring slower than anyone else. And that had a direct impact on the quality. For example, decay. Decay is that those fruit and vegetables, or in this case, these blueberries, that developed a fungal disease, in this case, botrytis cinerea. Control that had normal air went through the roof, followed by control atmosphere. But you can see here how the pathogen was also affected by that graduation. So, an interesting point. If we look at weight loss, we saw that there were no disadvantage in using graduated control atmosphere because it shows similar results compared to CA. And firmness, interestingly, showed better results, better maintenance of the firmness of the blueberry when we were having this, allowing this adaptation to the environment. If we look at ascorbic acid, that is one of the organic acids, vitamin C, that the consumer is interested in, but also is one of the substrates for respiration. Graduated control atmosphere was able to maintain higher levels or very similar to CA, and again, always higher than control. So a slightly advantage, no disadvantage. So at the end of the day, what I'm trying to, this is just a small case study. We can go in depth through this if you like, but in 15 minutes is really difficult, but I'm happy to have a chat with any of you into the details with that. But at the end of the day, it's really clear how the ripening and senescence processes are really linked to the respiration regulatory role. And in order to slow down that processes of ripening and senescence, the key element to look at is oxygen availability, because it is linked to ethylene sensitivity. It's another whole world that we can talk about. And that's what we are trying to look at now. Part of my research focuses now on how post-harvest responses linked to low oxygen availability permit or allow lower ethylene sensitivity. That is the ripening hormone, the hormone that triggers these changes, that are degradation changes. Okay? After that study, we've done this very similar experiments with other model crops, tomato, avocado, that is a very tricky fruit, because I don't know if you know, they only ripen once they have left the tree. So it's a nightmare of a fruit to work with, but very interesting. Cherries that traditionally have been thought to be non-climateric, so that they don't react to ethylene, but they actually do. And strawberries. So this is something that are being published at the moment, and we are looking at, in all these studies, also at the molecular level. Okay, so what are the genes that are actually involved or being activated or deactivated in these processes? And what we are trying to bring is this understanding of these physiological responses, this knowledge, into an applied field. So once we understand these changes, what we do is then going into developing this technology that targets specifically those changes. So we don't do anymore this trial-error situation, but we target at the specific response. So we have, I lead the agri-food strategy for Center of Excellence in Rwanda, Kenya, India, and we are trying to bring this knowledge and this technology to other countries too. I would like to thank Gatsby Foundation for inviting me here today and share the research with you all. I'm really happy. BBSRC and Johnson Matthey for supporting this research. My colleague, Professor Leon Terry, and one of my students, Tiana Miklo, who were involved in the research, and obviously my research group, the Plant Science Laboratory at Cranfield. So yeah, thank you very much. And yeah, any questions, I'm happy to answer.